Okay, very good. Well, thanks a lot, Ben and Tim, for the organization, and good afternoon, good morning, or good evening to everybody else. It's my pleasure to present CAMPI 6, which is on integrated multi-omics. Okay, so what is the vision and what are the objectives of this? So as you see depicted there, what we define as multi-omics in the context of microbiome analyses are, of course, metagenomic, metatranscriptomic, metaproteomic, which is the focus of all of our work, as well as metabolomic analyses. And so in relation to CAMPI 6, the vision is to establish robust, globally accepted best practices for metaproteomics through systematic benchmarking and integration of multi-omic data. The linked objectives are fourfold. The first is to define standards for multi-omic data generation. The second is to benchmark multi-omic pipelines from sample prep all the way to data integration. The third is on driving towards reproducibility and comparability across microbiome studies. And then fourthly, to explore the benefits of integrated multi-omics in the broader context of what we're trying to achieve here, not least, as already have been explained to everybody in the context of the grand challenges. Okay, so why this CAMPI? I think what we all recognize is, of course, a growing number of microbiome studies, but also within these studies, we see a growing complexity of basically the research questions that are asked, how they are addressed, and also what are the linked tools. But at the moment, what we are very much lacking, and you've heard now a lot about the individual CAMPIs, which are, of course, about standardization in metaproteomics, but also this applies to all of the other omics more broadly. So we don't really have standardized workflows for multi-omic analysis of microbiomes in general. And so that leads us to the need, which is for basically having coherent data integration and that allowing us to draw reliable conclusions. So again, as I already alluded to on the previous slide, what we are really needing are standardized workflows going from sample taking to cell lysis, to then biomolecular extractions, and then the subsequent systematic analysis of the resulting multi-omic data. Okay, so that leads us then on to, for CAMPI 6, the project idea. So in relation to the scope, we aim to compare existing data sets, so already published or potentially data sets that you have already generated, but then based also on an analysis of those data sets to design new studies. And so this is why we are proposing here a two-pronged approach. The first is indeed focused on the analysis of existing data sets for benchmarking. And then secondly, to use the knowledge that we've gained through those efforts to generate new data sets using defined best practice acquisition and analysis methods. So in relation to the key focus areas, there are three listed. So we have first and foremost, commonalities and differences across omic layers with a focus on metaproteome, so that you see somewhat illustrated here in the figure on the bottom, where we would aim to systematically, of course, look through the different omic layers and look at, for example, whether there are associations between metaproteome, metatranscriptome, and then, of course, for some genes there will not be, and what are the distinguishing factors for those, etc. So really to be able to do this in a coherent and systematic manner. And linked to this, of course, is to assess the information content of all of the different omic levels. That will, of course, be different, but there will be, of course, again, commonalities and we want to relate those then to, in fact, the differences that we see. And then as we all have this very strong interest in the functional dimension of microbiomes, we are aiming to, of course, use this integrated analysis across omes to really gain new functional insights into microbiomes, not least in the context of protein expression. Okay, and then the last key focus area here is really on the data integration. So what additional aspects can we cover through the integration of the multi-omic data, and how does that move us really beyond a single ome? Okay, so what are the expected outcomes of this? So these are fourfold. The first, and I've already alluded to this in one way or another, the first is on best practice guidelines for integrated multi-omic workflows. Second is to have standardized frameworks for, in particular, the analysis of integrated multi-omic data. The third is best practice, again, for integration of additional other omics data, so on top of, in fact, the metaproteomic data, for example, and also integration with metadata. And then the fourth one is, as an outcome, is to have been able to do really detailed comparative analyses across different microbiomes. And based on existing data in particular, we would see the foci here in 4 different microbiome areas. So first being human, then animal, environmental, and also engineered, for example, bioenergy, specifically biogas. So in relation to the project timeline, and this is, I have to say, really quite ambitious. We have to see in how these 3 phases will be achievable. So phase 1 of this particular CAMPI is on benchmarking existing data sets. So here, this is in fact also a call to the entire community. We are dependent on you to share those data sets with us. Please make us aware of, in particular, published data sets. So we aim this to take 6 months. Phase 2 is about the design and acquisition of new data sets, which would last then for the subsequent 12 months. And then finally, a third phase on integration and global dissemination, which would last then for an additional 6 months. So this would be leading to an overall timeline of 2 years, which I think in this context is really quite ambitious, but is achievable, I would think. Okay, and then what the general impact of this should be is also, again, along the lines of the grand challenges that were earlier alluded to by Daniel. We also see this very much contributing ultimately to a global microbiome multi-omics project. Also resulting from this will be harmonized protocols that will enable cross-study comparability. And finally, what we are aiming to get at is to really make novel discoveries in microbiome science, not least, for example, in the first instance, to have maybe some of these omic layers to contribute to, for example, models that potentially would lead us to mechanistic insights that in turn hopefully will then lead us to new discoveries. Okay, so this is in essence a call to action. So we would really appreciate your input, your contributions, not least in relation to submitting data sets, methods, and also your expertise. You can contact either myself or Dirk. We will also, of course, have the subsequent breakout where we can discuss this in more detail. And yes, we're very excited about this because I think it will allow us also to bring in other omes and to see how those are relevant for our interpretation of metaproteomic data, and then ultimately should lead us to novel understanding and novel insights into the functional roles that microbiomes play in the different habitats and environmental settings where we find them. All right, with that, again, thanks very much, and I look forward to the subsequent discussion. Thank you.